Okay, folks, boy, it's not very pretty outside this morning, is it? But I suppose the good news is on Thursday, the last time that we'll be meeting for a lecture, we can look forward to some good Christmas weather. It'll be snowing, is what the forecasts are calling for. So we'll end with a bang. Here are some more details for you about that optional review session that's mentioned in the syllabus and that I mentioned at the end of last week. The deal is that we will be in 116 Health Sciences for that optional review session 6 days from now, next Monday. And so Health Sciences is a building that I suspect many of you have not been in yet. It's kind of next door to us in that direction, right? It's a pretty cushy room. It's like a movie theater, more of a movie theater than this room is. So the plan is I'll get there probably a little before 10:30, and I'm just going to answer any questions that people throw at me. If you ask me what the answer to the fourth question on the exam is, the final exam, and I happen to know, I'll tell you, right? So anything I've got in my head, I'm happy to share with you. I'm not going to have a copy of the exam with me, partly so that I can honestly say I don't remember what the answer to the fourth question on the test will be. But give it a shot, and we'll see what we can do. Anything you want to ask about, I'll be happy to answer as best as I can. And I don't mind answering the same question multiple times. So again, starting a little before 10:30, if you like. And I need to be across the street at noon. So the syllabus says the review session will go till noon. I'm hoping I can cut it short at about 11:45 so that I can make it to another thing I've got scheduled for noon. Okay? Optional, come if you like. Don't feel like you have to. It's supposed to be an open spot in the final exam schedule. I don't think you'll have any conflicts with tests during that time. There won't be any recordings made that I'm involved with, right? If you guys want to record it, you're welcome to. But again, it's optional. Come if you like. I'll help as best as I can. And then, for the grand finale, there will be a final exam for this class. It's been shouted out in the syllabus. I've been mentioning it for the last couple of weeks. The final exam is on Tuesday of next week, so a week from today. Please notice that the final exam starts earlier than the class normally starts, right? I don't have control over this. It's determined by the registrar literally years in advance. But the final exam will be here in this room if you're, well, we'll have the final exam here in this room. The final exam starts at 8. If you're part of the online version of this class, so if you're in section 90, there won't be enough room for you here in this room, 120 Med Sciences. What you should do is instead go to 101 Fawcett Hall like you did for the first two tests. All right? So students in section 90, go to 101 Fawcett Hall. Everybody else come here. The exam's from 8 until 10. I fully expect that the average length of time that people will spend on the final exam will be in the ballpark of 45 or 50 minutes, somewhere around there. If for some reason you come late, say you stroll in at 9 o'clock or so, you know what's going to happen? I'm going to give you a test, all right? So whenever you get here, you'll get a test. When you're done, just leave, go home, take another final, whatever you got to do, but there's certainly no lecture or anything like that. So what I'm telling you is that any time between 8 and 10 o'clock, you can take that test. After 10 o'clock, we're going to get them graded, and I'm going to try and have final grades assigned sometime mid-afternoon on Tuesday the 13th, right? So don't be late. Not much point in being early. But anytime between 8 and 10 o'clock on Tuesday, you can come and take the test. Excuse me, there are going to be 58 multiple choice questions for you. 8 of those questions are cumulative questions, meaning that they will have come from parts of the first and second portions of the class, things that led up to the first test and things that led up to the second test. If you want a good guide in terms of what the final exam would look like, look at the exams that are available to you on the pilot page for the course. All right. If you haven't started looking at those exams on the pilot page for the course, the old versions of the final exam, what are you waiting for? Do it. Go check those out. It'll help you a lot. Okay. Back to work. I suppose before I get back to work, let me also mention one other thing. I've been getting email reminders that you can now start to evaluate the course online, right? So student evaluations of instruction, as they call it, are something that you can start to do. I think you've probably been getting email reminders about that as well. I want to really encourage you to take some time to give me some feedback about the course. I keenly read those evaluations when they become available to me. I think you probably have heard I don't get to see that sort of thing until long after grades are assigned. It's not going to have any effect on you for the course. But honest feedback is something that is really very helpful to me. If there are things about the course that you like, please let me know so that I can think if I might have ways that I can bolster that sort of stuff. And if there are things about the course that you don't like, please let me know, because I sure don't want this class to be just me talking as best as I can. I'm hoping that we can have sort of a dialogue and I can help you wrap your head around some of the things for the class. So good or bad, both let me know in those student evaluations. I'd appreciate it. And I'm really looking forward to hearing what you have to say. But for the business at hand, think about what we've been doing here in this class. One last sort of big overview picture, right? We started talking about some very basic chemistry. Excuse me, I tried to show you how it is those rules of chemistry applied to living things. And that's one of the reasons, the main reason why we pay attention to them in a biology class like this. We start putting molecules together, making biopolymers that could catalyze reactions. Got to the point where there were organisms that needed to pass on information from one generation to another, storing information in DNA, DNA replication, the whole central dogma. Wow, you know, it's quite a scope if you look back over the last month and a half or 2 months. Today, what I want to do with you is talk with you about how it is that molecular biologists actually figure those sorts of things out, particularly the things that we've been talking about for the last couple of weeks. And in a nutshell, in a couple words, what I want to talk with you about are best described, I would say, as the tools of molecular biology. Okay? I'm a molecular biologist. That's, you know, my PhD is strictly in biochemistry, but I got it from a molecular and cell biology department, and so I can kind of lay claim to the idea that I'm a molecular biologist. The research that I did for my biochemistry PhD was all molecular biology stuff. What molecular biologists do is they extract information from molecules, right? Kind of goes with the name. You know, biologist who focuses on molecules. And to really focus on molecules, you've got to have some tools that let you extract information from molecules. I think it's pretty fair to say that back when I got a PhD in molecular biology, and even still today when people get PhDs in molecular biology, what we're talking about here is an apprenticeship where you're hanging out with somebody who knows how to use the tools of molecular biology, and you learn how to use the tools of molecular biology by hanging out with a guy who's a master at the craft, right? In a very real sense, it's not that different, I think, from being a carpenter. Carpenters have a certain set of tools that you learn how to use, and it's a pretty small number. I don't know a lot about carpentry, but what I do know gives me the impression that carpenters can do some pretty amazing things with a pretty small number of tools. I'm talking about a hammer, I'm talking about a screwdriver, I'm talking about a lathe and a saw, right? What other tools do carpenters use? There's certainly others, but it's not like there's 50 other tools that a carpenter routinely uses. There are those 4 big ones. You probably could add on another 4 or 5 beyond that. But it's a fairly limited set of tools that they've been using for thousands of years to do some pretty cool work. Molecular biologists, good news, we've got at best 6 or 7 tools in our toolbox. And with that really small number of tools, we've been able to figure out all the stuff that we've talked about for the last couple of weeks. The central dogma, the structure of DNA, how genes get turned on and turned off. Remarkable stuff, remarkable insights from a very small number of tools. So the good news is there's a small number of tools. The good news is that what I'm going to talk with you about today is going to tell you, I'm going to go down the list. Here are the tools molecular biologists use, and check it out. If you didn't have this tool, you wouldn't have been able to learn this, or you wouldn't have been able to do that, right? That's the game plan for today. To be a really good molecular biologist, the trick is knowing what your tools are good for, but even more so knowing how it is you can combine 2 tools or 3 tools together, right? That's where the really clever molecular biologists make their money, is by knowing their basic set of tools, but also figuring, gee, if I used the hammer and the saw at the same time, check it out, look what I can do. And I'll show you a couple of examples of that during the course of today's lecture as well. And then, for the big finish on Thursday, I'm going to talk with you about genomics, what it is we now understand about the sum total of the heritable information of organisms. We'll talk about some prokaryotes, but ultimately I want to talk to you about what's going on with your genome, impressive as it is. But again, for today, I want to talk with you about the tools of molecular biology. If you like, you could say that these are the things that let people do genetic engineering, manipulating things at a molecular level so that they are suiting human purposes. Pretty cool stuff. Okay, this is just a reminder that last Thursday I talked with you about eukaryotic genes and regulation of gene expression. I don't feel I need to say anything more to you about that than what I said on Thursday. And I want to just jump right into this idea of cloning. So let's talk a little bit about cloning. This is one of the tools of molecular biology. Excuse me again. Now, when I say cloning, these days I suspect you guys are thinking about Dolly the sheep. Okay? And I'm going to talk about Dolly the sheep in a little while. That's a kind of cloning, for sure. But when a molecular biologist or a genetic engineer talks about cloning, they're usually talking about it in a much less dramatic sense. Cloning, strictly speaking, is getting lots of copies of a particular thing. All right? That's the essence of what's going on with cloning. And I said thing, getting lots of copies of a particular thing. I said thing because I wanted to leave open the possibility that we're talking here about lots of copies of a DNA molecule, but also the possibility of lots of copies of an RNA molecule or lots of copies of a protein molecule. So cloning, in some sense, could be used to describe any of those things. Typically, when a molecular biologist talks about cloning, we're talking about lots of copies of a chunk of DNA, a specific chunk of DNA. Here's why we need lots of copies of a specific chunk of DNA. It boils down to this. We're big, clumsy, oafish organisms, at least in the sense of talking about molecules. It's hard for us to manipulate a molecule or two. We really can only start to have some success manipulating a molecule when we're talking about manipulating a whole bunch of them, a big pile of those molecules. So we need lots of copies of a DNA molecule for us to be able to figure out what's going on with those DNA molecules and for us to be able to effectively change those DNA molecules. And that's what cloning is all about. So in the good old days of the 80s and 90s, when cloning was really starting to take off, here's what would happen. Is folks would say, gee, wouldn't it be swell if I had a whole bunch of copies of a particular organism's gene for, you pick it, whatever gene you like. Typically, it was somebody would say, I want a whole bunch of copies of the rabbit beta-globin gene, or I want a whole bunch of copies of the human beta-globin gene. It was often globin genes, it was often rabbits, it was often humans. How do you get a whole bunch of copies of a specific gene? Well, cloning is the answer. And the way that cloning was and still is done is you'd do something like this. You'd get yourself a bacteria, and do I need to tell you what bacteria? It's E. coli. Why are they, being politically correct here and calling it a bacterium? They used E. coli. We still use E. coli for these purposes. Remember, for E. coli, you got a big old circular chromosome in there, right? Contains all the genetic instructions you need to be able to do glycolysis and make ribosomes and the stuff E. coli needs to do. And as we talked about, I think a week ago today, a lot of times you'll find that bacteria, like E. coli, also have small extra chromosomal bits of DNA called plasmids associated with them. And what you can do is you can grow up a whole bunch of E. coli. And when I say a whole bunch, I'm talking here about liters, sometimes hundreds of liters of E. coli, big fermentation tanks. Grow up a bunch of E. coli, bust them open, isolate the DNA, and among the DNA that you would isolate would be the plasmids. And what you could do is you can take this plasmid, you can cut it open, so make it a linear molecule as opposed to a small circular plasmid, make it linear, and paste in between the two ends of the plasmid some gene of interest from some other organism, or not even a gene necessarily, some piece of DNA from some other organism, and then take this, and it's called now a recombinant DNA molecule, a recombinant plasmid. Take that recombinant plasmid, shove it back into a bacteria. Again, let's not dance around the bush, it's E. coli. And you end up with a bacteria that has a genetically engineered plasmid inside of it. Okay? So that's the essence of what's going on with cloning. And now what you can do is you can grow up liters, tens of liters, hundreds of liters, if you like, of this bacteria, bust it open, and have in your hands, literally in your hands, trillions of copies of that plasmid, and within that plasmid is that chunk of DNA that you were interested in. All right, and so that's the name of the game. That's what we're after here. We can grow these bacteria up a lot, and when they grow, we're going to each time get more copies of the plasmid and its chunk of DNA that we want to have in hand. And so you wonder, what are you going to do with that pile of copies of that chunk of DNA? Here, this slide's showing you some broad categories of things that you might do if you had lots of copies of a particular chunk of DNA. You may be able to sequence that chunk of DNA, determine the order in which the G's, A's, T's, and C's are arranged, and that might give you some insights about evolution and the relationship perhaps between pests and things that you spray on crops to kill insects, right? You may get some interesting insights that way. You may be able to just take your bacteria, let's say that chunk of DNA that you put in, codes for a protein that lets a bacteria, like E. coli, metabolize unrefined petroleum, right? Stuff straight out of the ground. And then what happens the next time you've got a big oil spill from a tanker that's spewing out, unrefined oil into the ocean, all you have to do is sprinkle some of those bacteria on the oil slick, and the bacteria naturally now start to chew up those petroleum molecules and clean up the mess for you. Wouldn't that be swell? That's something that can be done. That's something that is done now when there's an oil spill, is you can put bacteria to work, these genetically engineered bacteria with their recombinant plasmids, to be able to break down some of those compounds and reduce the problems that come from that spill. But you could also have your bacteria, remember, bacteria are using the same genetic code that you and I are using. It's a universal genetic code. And remember, what you can do then, because of that universal genetic code, is you can get the bacteria to make copies of a protein for you. So say you've identified a human condition, a human disease, that is rooted in that human's inability to make a particular molecule. Something like dwarfism, something like diabetes, right? Dwarfism a lot of times boils down to a paucity, too low a level of human growth hormone, a particular protein. You can have E. coli make the human growth hormone protein for you by genetically engineering a plasmid and getting the bacteria then to make the protein, busting open the bacterial cells that have made the protein, isolating the protein, and then giving it to somebody who doesn't have enough human growth hormone, and you've solved their problem. Diabetes, typically a problem with insulin. Not enough, more often than not. And what you can do is you could have bacteria grow insulin or make insulin protein for you. So they become little biochemical factories that can help you solve problems like dissolving blood clots and giving growth hormone to treat people who have stunted growth. So these are things that you can do with these cloned bacteria. You couldn't do cloning. And so cloning is itself, fair to say, one of these tools of molecular biology. And there's stuff that you can do. Once you've been able to clone something, there's stuff you can do because you used that tool. But you can't make clones unless you have some other tools to help you along. And so the first tool in the toolbox for molecular biology is a tool that's still used today. They're called restriction enzymes. Okay? Now, you guys in lab last week made recombinant plasmids. And you guys in lab last week used restriction enzymes. Let me tell you some of the stuff under the hood that may have gotten past you during lab last week. What's going on with restriction enzymes is pretty simply this. Bacteria, and I mean that, I chose that word carefully, bacteria have within them proteins that allow them to recognize foreign DNA and cut it up so that it gets degraded and doesn't cause the bacteria a problem. Okay? That's what restriction enzymes are ultimately all about from the perspective of a bacteria. And each bacterial species has its own suite of restriction enzymes, typically 2 or 3, that patrol around inside the cell, always looking for foreign DNA. Now notice, they can't be chewing up their own DNA. That's a bad thing, right? And so a lot of times bacteria that have these restriction enzymes will also have things in place that mask or shield their own chunks of sequence that these restriction enzymes recognize. But when a foreign piece of DNA comes in that hasn't gotten that protection, it gets broken down. And what the restriction enzymes all do, again, different species will have different proteins doing these tasks. There are on the order of 200 that you could buy online. In fact, I think you can get them from Amazon.com now. I haven't checked, but there are companies that make, that produce restriction enzymes for sale to molecular biologists. There are like 200 different restriction enzymes you might buy. What these proteins do is they look for specific tracts of sequence, and when they find them, here, there's a restriction enzyme, the first one that was ever characterized as EcoR1. E for Escherichia, Co for coli, Eco, R for restriction enzyme, Roman numeral 1, it's the first one in E. coli that was discovered. EcoR1 is a protein that E. coli makes that scans along DNA and looks for the sequence GAATTC. When it encounters that sequence, that's called its restriction site, when it encounters that sequence, the protein introduces a double-strand break in the DNA molecule. It breaks a phosphodiester bond between this G and this A, and between this G and this A. and it ends up converting this here big linear molecule into two smaller linear molecules. And you can see they're drawing your attention to the fact that there's a little bit of single-stranded DNA on both of those ends. They're sticky ends, sticky in the sense that they can base pair and stick with each other through hydrogen bonding. Okay? So E. coli makes this protein, EcoR1, that breaks up DNA molecules whenever it encounters that sequence. So that's a restriction site. Notice something subtle about this restriction site. It's a palindrome. Okay? I talked with you a little bit about a week ago about palindromes when I was talking about how DNA binding proteins work. You might recall EcoR1 is a DNA binding protein. It's a dimer. There's two proteins that come together, and each of the proteins lays across the major groove of DNA in a way that has it looking at a couple of nucleotides. Here, each dimer, each protein in the dimer, is looking at 3 nucleotides. And that's why it's reading the same forward as back. That's what a palindrome is. So what I mean is that if you read it from 5' to 3' in this direction, it's GAATTC. And if you read it from 5' to 3' in this direction, it's GAATTC. It reads the same forward as back. And there's this axis of symmetry in the middle here, such that the GAA, it's got a mirror image beneath it here of AAG. Palindromes. Reads the same forward as back. I can't resist telling you my favorite story about a palindrome right now. It's from an old Simpsons episode, and I do mean an old Simpsons episode. You might, if you watch the show, you might recall Principal Skinner at one point is telling the bus driver, you know, he's telling Bart, well, that's one palindrome we aren't going to have to worry about anymore because the bus driver just got fired. The bus driver's name is Otto, right? And so all that the Principal Skinner says to Bart is that's one palindrome we won't be talking about for a while. And everybody has to figure out what a palindrome means, I suppose. But he's referring to Otto, whose name is spelled O-T-T-O. It reads the same forward as back. So highbrow humor within The Simpsons, if you care to look for it. But that's a palindrome, just as this restriction site's a palindrome. So EcoR1. A restriction enzyme. Restriction enzymes are often described as being molecular scissors. They scan along DNA molecules, and where they encounter that restriction enzyme site, they cut like little scissors, and they make these sticky ends. And if you use the same restriction enzyme to cut up some other organism's DNA, you're going to get that other organism's DNA in fragments, where each of the fragments has the same sticky ends as the bacterial fragment or the plasmid fragment has. And the gray molecule's sticky ends can base pair and stick to the green molecule's sticky ends, and you could end up with something that is a hop, skip, and a jump away from being a recombinant DNA molecule. So the hop, skip, and a jump you need to do is you need to get another enzyme involved called ligase. And all that ligase is going to do is it's going to remake the broken phosphodiester bond here between this G and this A, between this G and this A, between this G and this A, and this G and this A. All right, so another enzyme comes in, heals up the breaks, and what you're left with is a recombinant DNA molecule. And so, seeing how this all comes together now in the context of cloning, and in the context of what you guys did in the lab last week, is you can start off with something like a plasmid. Here, the plasmid has already been engineered to have the ampicillin resistance gene associated with it. So they're showing that to you in a different color, right? But that's going to help us make sure that the bacteria not only get the plasmid, but keep it. Because if you grow bacteria up in the presence of ampicillin, and they lose this plasmid for some reason, they die, right? And so having the plasmid have the ampicillin resistance gene is a way that we can now make sure that any bacteria that got the plasmid keeps the plasmid, because otherwise they'll die. They won't be resistant to ampicillin. And it's also been engineered such that the beta-galactosidase gene is present on the plasmid. The beta-galactosidase gene, we've talked about it as the LAC-Z gene. It's the gene that allows bacteria to metabolize the sugar lactose. And so the LAC-Z gene, the beta-galactosidase gene, is there, and it has been engineered such that it's got the restriction enzyme site in the middle of it, an EcoR1 site in the parlance that I've been using so far today. So you take this plasmid, you excuse me, cut it with EcoR1, and you linearize it. That presumes, of course, that the restriction enzyme site is only at one spot on the whole plasmid, right? But if it is, you can make a linear version of the plasmid. You can, I don't know why, but you could take a hummingbird and grind it up and get its DNA, right, from its cells, and then cut it with the same restriction enzyme. And you can mix these hummingbird chunks of DNA with these linearized plasmids, use your ligase to seal up the breaks, and you can make a whole bunch of different versions of recombinant plasmids. Here they're showing you just 3 pieces of hummingbird DNA, but if you treated the hummingbird genome with EcoR1, you'd get many millions of different fragments of DNA, and you could clone every one of them. And you'll also get some that don't get any foreign DNA. The circular plasmid has just come back and recircularized. And what you can do then is get these plasmids into some bacteria. Let's call it what it is. It's E. coli. And you can grow those E. coli cells up on a plate. And if you have on the plate, just like you guys saw, if you have on the plate a chemical called XGAL, what will happen is that when the bacteria makes beta-galactosidase, it breaks XGAL up into something that has no color. Well, wait, that's the other way around. A recombinant plasmid with the disrupted LAC-Z gene can't break up. So if you cut XGAL, if you break XGAL with the LAC-Z protein, it makes a blue color. And if you're not making the LAC-Z protein, you grow a colony, but it has no color. Okay? And what we've got here then, it's called a screen. It's going to help us identify the bacteria that, the only bacteria that can grow on this plate are bacteria that are ampicillin resistant. So we know they all have plasmids. But this screen is going to tell us, not only does it have a plasmid, but is the plasmid a plasmid that has a chunk of foreign DNA interrupting the beta-galactosidase gene or not. If it doesn't have a chunk of foreign DNA and it gives you a blue colony, you chuck it. You got no more need for that guy. But for the white ones, the ones that do have some foreign DNA interrupting the beta-galactosidase gene, that might have a chunk of hummingbird DNA that you're really interested in. And so you collect all of these white ones, and you make yourself a library. Here you've got a 96-well plate that's got in each well of this 96-well plate a different colony. You would think each one, because there are millions of different fragments of hummingbird DNA that might have been cloned, you would expect, if you're only saving 96, that pretty much every one of those 96 would have a different chunk of foreign DNA. And you got yourself a library then that you can use to, with southern blotting, which I'll talk with you about in just a few minutes, you can use to find out where the chunk of foreign DNA is that you're most interested in. So that's one way to get an insert into your plasmid. Another way to get an insert into your plasmid might be to do this trick. What we're most interested in typically are eukaryotic genes, the things that are actually coding for what it takes to make a protein. And here's a shortcut that saves a lot of time. The approach I just talked with you about, where you're cutting up the whole hummingbird genome, that's going to give you introns, exons, 5' flanks, 3' flanks, the whole shebang. But if you just want to focus your attention on the protein coding regions, how about this neat trick? Crack open the cells, extract the messenger RNA, right? There are chemical tricks you can use that'll let you purify just the messenger RNA. Get the messenger RNA, use reverse transcriptase, which we get from retroviruses like HIV, make a DNA copy of the messenger RNAs. Now we call that a cDNA. It's a copy of the messenger RNA. And then clone those cDNAs. Now, when you're putting those things into your plasmids, all that you're going to be getting in your library are going to be the things that correspond to messenger RNAs. So you can have another kind of library. And when all is said and done, dim the light so you can really see how these guys pop. That'll wake people up a little too, perhaps, changing the environment in here. Check it out, what a little bit of knowledge can do. You guys see what's going on here? If you think that pig in the front looks a little bit strange, it is. That's a genetically engineered pig. And what's happened is that cloning stuff that we've been talking about, it works fine not just for bacteria, but we can do the same sort of manipulation of a eukaryote's genome. Pigs are eukaryotes. And what's happened to the pig in the foreground here is it's been genetically engineered through these restriction enzyme types of work to contain within it the gene for green fluorescent protein, isolated from jellyfish. And now, because pigs and jellyfish have the same genetic code, and because we know what sort of promoters are associated with genes that are expressed in pig skin cells, if you put that promoter in front of the coding sequence for green fluorescent protein, and then you take that construct and you slip it back inside a pig's cells, you can make pigs whose skin glows in the dark. And once again, I ask you guys, why aren't you working on making fluorescent roses or daisies or something like that? Because for goodness sakes, if you can do this sort of manipulation to a complicated organism like a pig, you should certainly be able to do the same sort of thing for flowers. And I think there's a lot of money to be made by making those moonlight gardens that literally are giving rise to their own light. So you can make transgenic organisms. So green pigs. There's probably not a lot of market for green pigs, right? But check out a more practical application of this kind of transgenic organism. A little while ago, I mentioned diabetes, all right? When I was in graduate school, it was big news that genetic engineering and recombinant DNA was going to solve the problem of diabetes. Right? Here's what the problem had been, right? For about 150 years, people had known that the problem that diabetics had is they typically didn't produce enough insulin, right? There are other ways you can be diabetic, but that was the typical one. And so for about 150 years, with that realization, you know how you treated somebody who had diabetes? You'd tell them, as a physician, you'd tell them, don't eat that much sugar. That's part of your problem. But if you wanted to intervene beyond just that sort of counseling, you know what you'd do? You'd give those people shots of insulin. And what kind of insulin do you suppose you would give them? Well, you'd give them the insulin that was most readily available to you, that you could get lots of, right? And where, before genetic engineering, did you get a protein like insulin? We've talked about this before. If you've ever been to a slaughterhouse, they literally have buckets, barrels full of blood, right? And so you'd go to a slaughterhouse, and insulin would come from the buckets of blood from pigs and from cows. They'd purify the protein, and they'd shoot people up with pig insulin. But the problem with pig insulin is that while it's very similar to human insulin, it differs in 2 positions. 2 amino acids out of 56 are different. And what would happen is over time, people, diabetics who are getting pig insulin, would develop an immune response to those subtle differences, to the pig insulin, to the point where the pig insulin became more of a problem than a help for those people. You were shooting them up with something that they were effectively allergic to, and they'd get all sick, and they didn't like that. So you know what you would do at that point? You'd switch over to cow insulin, right? Because you could get a lot of that, too. Problem with cow insulin is it differs from human insulin at 2 other spots, two other amino acids. And over time, you'd develop an antibiotic resistant, or you'd develop an immune response to pig insulin. You'd later develop an immune response to cow insulin. What's left? If you were lucky, you lived in a world, or a part of the world where there were a lot of horses, and they would be able to give you horse insulin. But after you'd gone through pig, cow, and horse insulin, there's only one alternative, aside from death, which was not considered to be a viable option, literally. That was sort of a joke, I suppose. The only other option was human insulin. And where did you get human insulin from? Dead folks, cadavers, right? And that was pretty much the end of the line, limited supply, a lot of people. Yeah. So it was bad news, right? Well, check it out. When I was a graduate student, for the first time, it became possible to clone the human insulin gene and put it into E. coli, into a bacteria, and grow up in big fermentation tanks, effectively an endless supply of human insulin. And you didn't have this problem anymore about the mounting an immune response to the pig, the cow, and the horse insulin, and you didn't have to harvest insulin, human insulin, from cadavers. Problem solved. What could be better than that as a solution? There's something better. Let me tell you what the better thing is. In the last 5-10 years, you know what's happened? Is the companies that had been growing the bacterial human insulin from the clones shut all those fermentation tanks down. They're using them now for other purposes, like human growth hormone and other enzymes. You know where human insulin comes from now? If you're a diabetic, you know where your human insulin comes from? This is so cool. Where it comes from now is genetically engineered cows, right? And the way that these cows have been genetically engineered is really cool. We've taken the human insulin gene, right? Codes for the human insulin protein. We stuck a promoter in front of it that causes this gene to be expressed in one specific tissue within cows, right? And you guys know how all that works, right? We've talked about that just last Thursday. How it is that one promoter might cause a particular gene, a transcription factor, binds to that promoter, and that transcription factor is only available in one tissue. Guess what tissue they have the human insulin gene being expressed in cows? Their udders. Right? So the same promoters that are associated with the proteins that show up in cow's milk, now we put that promoter in front of the human insulin gene, and there's a herd of about 500 cows whose milk produces human insulin. And you milk those cows, and you purify the human insulin from it, and you got all the insulin you need for the entire world's population of diabetics. How about that? It's hard to imagine how it gets too much better aside from genetically engineering the people with diabetes themselves. And that's probably not too far down the line that there will be a way that you can infect them with a virus that inserts another copy of the human insulin gene and maybe gets them producing insulin again for themselves, and you leave the cows and the bacteria out of the equation entirely. So pretty cool stuff that you can do with transgenic organisms. All right, well, funny thing. It's not, oh, there it is. Funny thing is that cloning became very passé, or better, restriction enzymes, and that kind of recombinant DNA technology became kind of passé in the mid-90s, and it got replaced with something For the most part, it's been replaced. It's still used for certain purposes. But there's a new kid on the block since the mid-90s called the polymerase chain reaction. Good story associated with the polymerase chain reaction. The fellow who discovered PCR, the polymerase chain reaction, his name is Kary Mullis. He got a Nobel Prize in the early 90s for figuring this out. Kary Mullis is a very colorful character. Look him up on Wikipedia, Google him. Be careful if you look at his websites, they're kind of X-rated, right? But Kary Mullis is a very colorful guy, discovered PCR by his own account. He discovered PCR while he was tripping on LSD, right? He was having some sort of weird feverish dream, and he had just been thinking about snakes, and the snakes were coming back, and the head of a snake was biting the tail of another snake, and he saw this sort of cycle, and then he was thinking about DNA, and PCR was delivered to him in this process. So he gets a Nobel Prize, you know, pretty open about his use of illegal drugs and narcotics. And then once he gets his Nobel Prize, you'd think this is a guy, you heard about Watson and Crick, they made their careers off of that. Didn't work out that way for Kary Mullis. He's pretty much abandoned science entirely. He's no longer doing anything associated with science. The last that I heard is that he had taken up a hobby of photography, right? And, you know, it's kind of artsy stuff. It's like black and white photography, not normal photography. And it's not just black and white photography. It's black and white photography of naked females. And it's not just naked females. It's naked prepubescent females. And so there's a child pornography sort of thing going on here with Kary Mullis. It's kind of weird. But nonetheless, he comes up with this cool idea of PCR. And everybody who's a molecular biologist these days uses PCR. And what PCR does is it allows you to choose regions that you want to make lots of copies of, and you can do it all in a test tube and leave the bacteria out and the cows and the green glowing pigs out of the equation. You can do it all in a test tube because it's entirely an enzymatic process. And it more or less is a copy of the way that bacteria, and eukaryotes as well, copy their own DNA. So this slide shows you the PCR process in all of its glory. Let's break it down bit by bit, and I'll show you how PCR works. I'll tell you, at first pass, when you look at PCR, it looks kind of confusing. I remember, and you look at it and you think, oh, this is complicated. I don't know that I want to figure it out. But I want to encourage you to buckle down, and even if you don't understand what I'm talking with you about for the next 5 minutes or so, look at these pictures and wrap your head around it. Because what's going to happen is it's going to get you really comfortable with how DNA polymerases work, how it is that DNA itself gets replicated, and it's going to give you a good insight in terms of a universally used tool of molecular biology, right? Molecular biologists all use PCR. And if you take the sophomore level class in the series here at Wright State, there's a laboratory component associated with it. You're going to be doing lots of PCR, and you're going to have to figure it out then anyway. So here's what's going on with PCR. You got your big squiggly piece of genomic DNA. There's lots of stuff here that you could care less about, right? But there's this little stretch here that's shown in blue that you're really interested in. Okay? Could be that it codes for a particular protein. It could be that it's something that's very likely to be different between one person and another person. And it could then become the basis for a test that you would use to solve a crime, right? DNA profiling sorts of things. But again, there's this stretch of blue that you think is really interesting. It's mixed in with all this gray stuff you don't care about. And so you want to have lots of copies of the blue thing. What you might be able to do is this, is you could take that region that you're interested in and denature it. Separate the two strands. Go from double-stranded DNA to single-stranded. And you're not going to do it just for that blue region. All the DNA is going to become single-stranded, but let's focus our attention on what's happening for the region that you really like. You separate them through a denaturation step. And then into the mix, you add some primers, some short stretches of DNA, single-stranded DNA that can be synthesized in the lab. There are machines, you can dial up, I want to make single-stranded DNA, and I want it to be this particular string of 20 nucleotides in a row, or 25 nucleotides in a row, or 26, right? You can spell out to a machine and have it automatically synthesize for you a primer, single-stranded chunk of DNA. And when you're designing your primers, you want to make two. You want one that will specifically base pair with one end of the region that you're interested in, and you want the other to specifically base pair with the other end of the region that you're interested in. Now, I tossed out some numbers a little bit ago. I said maybe 20 nucleotides long. Oh, go crazy. Make it 25. Make it 26. All right? You need a certain length in order to make sure that that string of characters, that string of nucleotides, doesn't occur anywhere else within the genome. Right? You don't want it to be you don't want your primer to be just 4 nucleotides long, because a stretch that's only 4 nucleotides long, that primer is going to sit down all over the place. By the time you're at about 20 or 21 nucleotides long, the chances are pretty good that your primer will sit down at only one spot in the entirety of a eukaryote's genome. Okay? And we've done that for this primer, and we've done that for this primer. They're each going to sit down just on one spot in the entire genome, one at the beginning of the region we're interested in, one at the end of the region that we're interested in. And so you split your DNA up into single strands. You let your primers find their complementary sequences to each other, and then you let DNA polymerases do their thing. And remember, what DNA polymerases like to do is they like to sit down on some existing double-stranded stretches of DNA and then extend in one direction, 5' to 3', off of an existing stretch of double-stranded DNA. So this is just what a DNA polymerase is looking for. This DNA polymerase doesn't really have any interest in, but boy, you set a DNA polymerase on this and it's raring to go and make another copy of this stretch of single-stranded DNA. Extend this primer and make it into a longer stretch of double-stranded DNA. And check out what happens as a result. We started with one double-stranded molecule that was blue, one double-stranded molecule that we were interested in, and at the end of one round of PCR amplification, we end up with 2 double-stranded molecules of the region that we were interested in. Okay? If you denature, anneal, and extend a second time, you won't have just 2 copies of that region of interest anymore. Now you're going to have 4. And if you do that another time, go through another round of PCR, your 4 becomes 8. You do another round of PCR amplification, your 8 becomes 16, 16 becomes 32, 64, 128, 256, right? Typically, when you do PCR in the laboratory, you won't do just 1 or 2 rounds of PCR. You'll typically do on the order of 20. You end up with 2 to the 20th copies of the molecule that you're interested in. And when I talk with attorneys about this, because this is central to the way that DNA profiling works for shows like CSI and such, when I talk with attorneys about this, I'll often tell them that you go from looking at a needle in a haystack, right? A little tiny stretch of DNA in a vast pile of stuff you're not interested in. You go from looking at a needle in a haystack to looking at the end of these 20 rounds of amplification to looking at needles in a needle stack that has a little bit of hay mixed in. Because the regions that you're not interested in, those gray bits of DNA, they don't get replicated. It's only the blue parts that are getting replicated because of the way these primers are sitting down on complementary sequences. And before long, what ends up happening is that the copies end up being delimited by the primers themselves. If you, with paper and pencil, sketch out what's going on with PCR, what you'll find is this slide is correct. At the beginning, when the primers sit down, they just keep on going until they fall off the end of the world. But eventually, what happens is that if you make a copy of this molecule, it's going to have only the information that begins with that primer, and you start to end up with molecules like this one, where the beginning of the molecule is defined by one of the primers, and the other end of the molecule is defined by the other primer. Most of the products of PCR at the end of 20 rounds of amplification look like these molecules. It is just, that amplified DNA is just the region that begins and ends with the primers that you had initially synthesized in the laboratory. And what was cloning all about? It's getting in your hands a chunk of DNA that you can poke and prod and do stuff with. That's what PCR delivers to you. There's no longer the bacteria, there's no longer the cow, right? PCR is giving you all those copies. And this is showing you what happens at 3 cycles of PCR, 3 rounds of denaturing, annealing, and extending. But as I said, you'll typically, in a laboratory setting, do 20, maybe 30 rounds of PCR amplification. You get an enormous amount of copies, number of copies of the molecules that you're interested in. 1 round of PCR amplification takes about 2 or 3 minutes. And so you can do this whole process, 20 rounds of PCR in the span of a couple hours, hour and a half. Could typically be all that you need to get trillions of copies of a molecule that you're interested in. So an enormously powerful technique, very clever method that sort of gets around the need for restriction enzymes and even the need for transgenic organisms. But you could still clone your PCR products so that you could dial them up and look at them again later. All right, let me talk with you a little bit about another tool. So I'll tell you what, I think I've talked with you about 3 tools. I've talked with you about cloning, I've talked with you about restriction enzymes, I've talked with you about PCR. There's still a couple more to talk about. Let me talk with you about a guy's trick. Edward Southern, came up with this trick that we now call Southern blotting. Okay? And Southern blotting is essentially 2 tricks, 2 tools that get rolled into one. All right? So I've talked with you about cloning, I've talked to you about restriction enzymes and PCR. Let's talk about gel electrophoresis, and then let's talk about blotting or hybridization. So we'll get the tally of tools in our toolbox as molecular biologists up to 5. So Southern blotting starts with gel electrophoresis. And you guys are old hands at gel electrophoresis now. How many times did you do gel electrophoresis in the lab this year for Bio 1120? 2, 3 times at least. Here's what's going on very simply with gel electrophoresis in case you didn't notice when you were doing it in the lab. Is you make yourself did they have you guys use Jell-O? Cool, good, because you know they also I'm firm. I'm telling you guys have got to feed these people asparagus, right? If you look in the lab manual, there's an experiment where you check to see if you can smell a byproduct of asparagus metabolism in your urine, and the prep lab people have autocratically overruled me and said, oh, we're not going to give him asparagus. I was like, what are you talking about? Give him asparagus. So I don't know. It's a battle, a tension going on between me and the prep lab people about that. And I was afraid that you were going to tell me that they didn't do the Jell-O bit. But here's what goes on with gel electrophoresis. You make yourself a vertical slab of Jell-O. And Jell-O will work in a pinch. But to be fancy in molecular biology, we don't use Jell-O, we use agarose. It comes from seaweed as opposed to cows' and horses' hooves, right? Where you're getting the gelatin from for Jell-O, they use seaweed to get agar. Same idea, same principle, very similar molecule to gelatin, but you make agarose gels, you make a flat slab of these guys. There's wells, little divots within the flat slab. that you can put samples into, DNA molecules, for instance, that you've isolated, or maybe DNA molecules that you've generated through PCR amplification. You can put them into these wells, run an electric current through the gel, and separate molecules, DNA molecules here, but it works for proteins as well, and it works for RNA as well. You can separate molecules on the basis of their size. Big molecules have a hard time fighting their way through the gel. Small molecules can wiggle their way through further and move faster. You do size fractionation. Separate the molecules on the basis of their size. DNA is negatively charged, so it's naturally pulled toward a positive electrode, and it's naturally repulsed from a negative electrode. So you separate the DNA molecules through gel electrophoresis. That's actually one of the tools of molecular biology, gel electrophoresis. And you get to see stuff like this. Right here they've used a dye called ethidium bromide, which fluoresces in ultraviolet light, and you get to see where the DNA molecules have used. We have you guys use something different than ethidium bromide, and about as little as 5 years ago, we had the people in Bio 1120 use ethidium bromide. Turns out ethidium bromide is something that's called a teratogen. Teratogens are chemicals that have been demonstrated to cause birth defects in developing embryos. If it's teratogen, not only is it bad for developing embryos, you can pretty much count on it being bad for people as well. And so we kind of ditched the ethidium bromide, and we had you guys use cyber green, as I recall, which is a little bit safer. But anyway, you can visualize where these molecules have separated out. But here's what else you can do. And this takes it beyond what you did in the lab. You can take your bits of DNA, here it's been DNA that's treated with EcoR1, size fractionate the DNA on a gel, right? 3 different lanes, each with a bunch of fragments in it. And then you can do Ed Southern's trick, Southern blotting. And I'm not making this up, right? The way that Southern blotting works is this. It sounds sort of like you might do it in your kitchen. That's exactly how it is that molecular biology labs tend to work, right? What you do is you take your gel, your agarose gel, and you guys have seen and worked with agarose gels. They're kind of wiggly and sloshy, not particularly stable or solid. What you can do is you can transfer the size fractionated molecules off of the gel onto something that's a little more permanent, a membrane. And the way you do that is you get I don't even know what to call this. What I'm envisaging is the sort of thing that we make Rice Krispie treats with at my house, right? You know, like a cooking pan kind of thing. You fill it with buffer, you put a sponge in it, and then you take your gel and you set it on top of the sponge. It's not washing around in the liquid, but you fill it with a buffer, a liquid. The sponge is in contact with the liquid and the gel, and then you put a membrane. And when I say membrane, you know what I'm talking about? I'm talking about a piece of paper. Paper will literally work in a pinch. And it looks a lot like a piece of paper. You put your membrane on top of the gel. The membrane, I said paper will work in a pinch. Here's a secret for you. If ever you need to make your own membranes and you don't want to splurge and buy them from a company that makes the membranes, the membranes are nitrocellulose. You know what nitrocellulose is? Cellulose is paper. Nitrocellulose is paper that's been treated with some nitric acid. You wash it through nitric acid a little bit and you make it nitrocellulose. I've made nitrocellulose before. As an undergraduate, you guys, okay, this is dangerous, so maybe you shouldn't do this. But as an undergraduate doing undergraduate research, I highly recommend do undergraduate research. As an undergraduate doing undergraduate research, one day I got the recipe for making nitrocellulose. Okay, oh, this will be fun. Let's see what this is like. And I took some paper and I put it into a bath with some nitric acid. And you also need to add a little bit of sulfuric acid to just juice it up a little bit. And you just set it there. You pretty much just rinse the paper through the bath of the nitric and sulfuric acid. Just rinse it through, dry it off, and you got yourself some nitrocellulose, a membrane, right? If you do a little more than a rinse, if you let the paper sit in there for a little bit, it gets more and more nitrocellulose-y, right? It gets even more nitrates onto the nitrocellulose. And you get to a particular point where, have you ever heard of something called gun cotton? In the old days, for flintlock rifles and muskets and things like that, you'd have cotton that was like mostly cellulose that would get treated with nitric and sulfuric acid so that when you had your flintlock, the spark would come, it would touch the gun cotton explodes, and then that triggers the gunpowder to go off and it shoots your shot wherever you want it to go. You let your nitrocellulose membrane in the nitric and sulfuric acid too long, and it goes from being a nitrocellulose membrane to being sort of like a membrane of gun cotton. And here's the thing that really surprised me. And this is not going to be in the test, right? Just a little side story for you. Here's the thing that really surprised me. There's never any warnings, never any cautions about this when you're looking online about how to make nitrocellulose. It turns out if you leave the paper in the nitric acid and the sulfuric acid, it becomes not just gun cotton, it gets beyond gun cotton to something that spontaneously combusts and explodes. And what you find is this membrane bathed in nitric and sulfuric acid under the water of the nitric and sulfuric acid that catches fire and starts spewing stuff all around. The stuff it's spewing, of course, is nitric and sulfuric acid. And it's really spectacular, but it's not something I would recommend. Yeah, it's kind of fun to know about. You don't want to actually repeat that experiment of mine, but I've had this personal experience. So I've made my own nitrocellulose. There's not that much to it. And back to the topic at hand, you take your gun cotton paper and you lay it on top of your agarose gel. And remember, your agarose gel is sitting on top of a sponge, and that sponge is soaking up buffer inside of a Tupperware container, okay? And then on top of your nitrocellulose membrane, and you use nitrocellulose because DNA sticks better to it than to just plain cellulose, by the way. It sticks to cellulose, it sticks better to nitrocellulose. You take your nitrocellulose membrane, and on top of that, you put some paper towels. And you know what paper towels we use in my lab and that we used when I was a graduate student? I should get, how many times have I done product placement here today? I've talked about Amazon.com, right? I've talked about, you know what paper towels you use? What's your favorite brand of paper towels? Of course you use Bounty. Why? Because it's the quicker picker-upper. It always has been. And what you want is you want to suck that buffer up through the agarose, up through the sponge, through the agarose gel, through the nitrocellulose membrane, into the stuff on top of it, the paper towels, and you put a weight like a book on top of it to help hold it all in place to make sure you get good contact throughout. But the thing is the DNA can't move through that nitrocellulose membrane. It sticks to the membrane, the buffer moves through, you pull it all through, the DNA sticks on the membrane, and when you're done, you throw away the book, or you sell it on a used book market, you throw away your paper towels, and you got your membrane in a squished agarose gel, you throw the gel away, you get your membrane, and on the membrane, wherever the DNA fragments had moved on the gel, now they're stuck to the membrane. And you take your membrane, and I'm sure there are competitive products out there, but what every molecular biology lab uses is a sealamine machine, literally a sealamine machine. You take your membrane, you put it into a plastic bag, you mix in some warm soapy water, as well as some fluorescently or radioactively labeled probe, and the probe could be the same sort of thing you used as a primer for your PCR a little bit ago. You take that single-stranded fluorescent or radioactively labeled primer, you wash it over your membrane, you seal the bag up, you let it slosh around for a little while, typically for an hour or two. You cut open the sealamine bag, drain off all the radioactive and fluorescent primer and soap, and you end up with a membrane that if you put next to an X-ray film will show you where the fragments of DNA that you were interested in moved on that original agarose gel. That original agarose gel, if this is a genomic DNA digest, is going to have a whole smear of fragments of DNA. Within that smear, there may be a couple of spots, fragments of DNA that correspond to what you were interested in. You can see where those fragments were relative to all the rest through this process of southern blotting. So, southern hybridization, southern blotting, that's a trick that we can use to help us identify where DNA molecules that we're interested in happen to be. It's rooted on the fact that complementary DNA sequences stick to each other. You can use it on agarose gels. You can use it on the genomic library that I showed you before, that 96-well plate. You can use the same approach to figure out which of those clones has the fragment, the gene that you're interested in. It's something of a digression, but not entirely a digression. Ed Southern is the guy who came up with this. A colleague of his happened to have the name Northern. All right? So southern hybridization is when you're talking about hybridizing DNA molecules to other DNA molecules. The same trick works really well when you're hybridizing DNA molecules to RNA molecules. That's northern hybridization. How about that? So Edward Southern comes up with DNA to DNA hybridization. That's southern blotting. Northern comes up with DNA to RNA hybridization. That's northern hybridization. Is there anything else that might, there's 2 points of the compass. How about Western? A guy named Western picks up where those two guys left off. And I'm not making this up. Western blotting is when you use DNA to hybridize to proteins that have been separated through gel electrophoresis. Pretty cool, huh? And so we got southern blotting, northern blotting, western blotting. Is there anybody in this class named Eastern? Because we got to come up with something for you to do. We got only 3 of the 4 points of the compass taken care of so far, but it's DNA to DNA, DNA to RNA, and DNA to protein. So we got to come up with something for somebody named Eastern to do. And so remember those 96-well plates I talked about before? You can transfer the DNA onto a membrane. Here they're using nylon as opposed to nitrocellulose because nylon's not flammable. It's much safer, right? You do the same sort of coelomere trick, right? And you can do just as we had talked about before. And the way the technology is worked out today is this is what's called a gene chip, the results that come from a gene chip. Gene chips are southern blotting on steroids, just taken to the extreme. A gene chip looks for all the world like the cover slip for a microscope slide. Little tiny, flimsy piece of glass. On those cover slips will have been spotted by a machine tiny dots that are, you can sometimes get 10,000 dots onto a cover slip, tiny dots of different fragments of DNA. And they typically correspond to cDNAs, so reverse transcribed DNA, but they'll be cDNAs, copies of all of the genes from a particular organism. That's what we're looking at here on this particular image. And what you can do then is you can take your gene chip that has all those different spots of DNA affixed to now, instead of nitrocellulose or nylon, to that little glass silicon chip. And you can wash across that fluorescently labeled messenger RNAs that you've isolated from a tissue. And you can look then to see where it is those RNAs from the tissue stick on the gene chip, and that tells you what genes are being expressed for this particular tissue. All right? Can you think of a practical application for that type of analysis? I'll give you one. Let's say you've got cancer, and you want to know what's different about your cancerous lung cells as opposed to your non-cancerous lung cells, what you can do is you can take a biopsy, get some of the non-cancerous tissue, and figure out what genes are being expressed, and then take some of the cancerous tissue and figure out what genes are being expressed. And you know what you find when people do that? It's typically just a difference of 4 or 5 genes being expressed in one versus the other between a cancerous and a non-cancerous tissue. And when you can figure out which genes your tumor is expressing or not expressing, that helps with characterizing what kind of cancer you have, what version of lung cancer you have, and it gives physicians some insights in terms of what treatments might work best for you. Maybe you can turn off some of those genes and thereby make those cancer cells less viable than they would have been otherwise. All right? And so we're on our way, and I'll talk with you about this a little bit more in class on Thursday. We are on our way to personalized medicine, such that the drug that you're prescribed is dependent upon what genes you're expressing under different circumstances, right? And these gene chips give us the ability to do that. All right, so there you go. There's a whole bunch of different kinds of hybridization. The last, and certainly not least, tool that molecular biologists have in their toolbox is DNA sequencing. DNA is an information storage molecule. We'd be in a sad state if we couldn't read that information. DNA sequencing is all about cracking into that information, determining the order in which nucleotides are arranged. It literally started out with a Harvard faculty member. Oh, here he is, Wally Gilbert. Walter to you guys. I know Professor Gilbert. But Wally Gilbert and his graduate student. Wally Gilbert has his background in organic chemistry, and his approach for sequencing DNA is very chemically based. It's very brute force, and nobody uses it anymore. But when he and his student first figured this out, they sequenced 50 nucleotides from the human beta globin gene. They were able to show that it was the human beta globin gene because previously others had figured out what the genetic code was, and previously others had figured out what the amino acid sequence of the human beta globin gene was, or the human beta globin protein was. And Maxim and Gilbert were able to show the first 50 nucleotides of the beta globin gene, when conceptually translated, gave you the string of amino acids that we knew were at the beginning of the human beta globin protein. All right, so that's what they did. They showed that they could determine, with this very brute force chemical approach to breaking up DNA molecules, they could determine the sequence of nucleotides. Those first 50 nucleotides of sequence literally got them a Nobel Prize, right? For figuring out how to sequence DNA, even with this clumsy brute force approach that nobody uses anymore, they get a Nobel Prize. That was in '76. As a graduate student myself in the late '80s, I was a prodigious DNA sequencer in my day. I cranked out, in my PhD thesis, I cranked out on the order of 65,000 nucleotides of sequence. Okay, I used the Sanger method for my sequencing. I'll talk with you about that in just a bit. 65,000. So in the span of, what, 20 years, 50 nucleotides gets you a Nobel Prize, 65,000 gets you a gold star PhD, right? My wife got her PhD something like 5 years after I got mine. As an appendix in her PhD thesis, she has 2 million nucleotides of sequence. The difference was clumsy chemical approaches at the beginning gave rise to manually done Sanger sequencing when I was working at generating lots of data, to automated processes that my wife used to generate the millions of nucleotides as the appendix of her PhD thesis. Nowadays, there are machines that will sequence an entire bacterial genome for you in the span of an afternoon. Millions of nucleotides in the course of an afternoon. highly automated Sanger sequencing. So I think I've got like 50 seconds left. Let's see how much damage I can do with 50 seconds, and then I'll come back and I'll talk with you on Thursday at the beginning a little bit more about Sanger sequencing. I'll tell you what, I'm just going to do damage if I talk with you about that right now. So let me just leave this slide hanging. At the very beginning of class on Thursday, I'll take you by the hand and tell you how it is Wally Gilbert broke up DNA molecules. Then I'll talk you how Maxine Sanger came up with a better way to do the process, and then we'll talk about genomics. And that'll be it. So see you guys on Thursday.